This talk is about convalescent plasma for the treatment of COVID-19. First, a bit about names. COVID-19 is named after coronavirus disease 2019, and it's caused by the virus, severe acute respiratory syndrome coronavirus 2, which you'll see referred to as SARS-CoV-2. It's called a coronavirus because if you look on the left-hand picture, you can see that the virus is surrounded by these spikes, which are thought to resemble a crown, hence corona. On the right-hand side, you can see each of those spikes is actually heavily coated with sugars, and those sugars in purple and yellow actually mimics human sugars and stop the immune system recognizing the virus. But the virus has to have one bit which attaches onto the cell, and that's that bit in blue. It attaches to the receptor on the cell, in fact, something called the ACE2 receptor, and this is the Achilles heel of the virus as far as antibodies is concerned. What are antibodies? Well, when one thinks about the immune system, there's two broad arms. There's innate immunity and adaptive immunity. Innate immunity is something that we're all born with, and we've got cells in our body which have got receptors on their surface which bind bits of bugs, and if they recognize those bugs, they then eat them and destroy them. But some bugs can get through that first layer of immunity. And there's also something called adaptive immunity. So that when you're born with millions of different forms of random molecules on the surface that might recognize a bug. And if they do recognize a bug, the body very cleverly makes the cells that make that particular random receptor grow and retain in your body for the rest of your life. And so you have got a memory of how to deal with the bug that deals with that is recognized by that cell. And there are two sorts of cells that have this ability to have a memory to recognize that bug. There's B cells and there's T cells. B cells make antibodies. Antibodies are molecules, and there's a picture here, which if you look at the blue bit on the left-hand side, every single antibody has that bit, and that acts as an adapter for the rest of the immune system to latch onto if the yellow bit on the end recognizes a bug. And if it binds to a bug, then the rest of the immune system can then eat it and destroy that bug. In fact, it's a lot more complex than that. There's lots of different types of antibodies, which you might want to ask me about afterwards. And here's a picture of a coronavirus with antibodies that are recognizing it and coating it. And that stops the virus replicating. It can stop, if it binds onto that blue bit that I showed you, it'll stop it attaching onto new cells and infecting new cells. But also, once the virus is coated with the antibody, the virus can be removed by different cells of the immune system. So, what's convalescent plasma? Plasma is the liquid portion of blood. So if you take all the cells out of blood, you're left with a straw-coloured fluid, and this contains the antibodies. Almost everyone who's been infected with any virus, actually, but including SARS-CoV-2, develops antibodies against that virus. A lot of those antibodies are pretty useless at actually preventing the virus replicating. For instance, if an antibody is directed against one of the internal bits of the virus, but some antibodies are particularly good, and if they bind that blue bit, and these are so-called neutralizing antibodies. And what we're finding from the donors which are coming forward to us is that only about 40% of the donors have got these neutralizing antibodies. The best antibodies we're finding are from patients who've been very ill. They tend to be older, they tend to have blood group A actually, and they tend to be male. Unfortunately, a lot of elderly males are not fit to donate afterwards, and actually we're finding that males are significantly less likely to donate plasma than females. I don't know why. We can speculate about that. So we are short of donors. So what we're doing in Scotland is everybody who's had a diagnosis of COVID-19 is being sent a letter, and they have to reply to that letter, and then they're contacted to see if they're willing to donate, and if they're fit enough to donate. A lot of the patients we're finding are not fit enough. Another problem we have is that the antibody levels decline relatively quickly, so that after about 2 months, about a half, 3/4 of all the donors don't have enough neutralizing antibody to actually make it worthwhile collecting. And when we collect that plasma, it's frozen down, and if patients come in with COVID-19, we can give it to them. So far, hundreds of thousands of patients have been treated in the USA, but it's just been given to them on the basis that it might be a good idea. They haven't done a proper trial. They have compared, however, people who've had high titer neutralizing antibodies, lots of antibodies, versus low titers. And it looks as though the people who've received high titer antibody, especially if they receive it early on in their illness, the death rate seems to be about 30% less. So it indicates it probably works, but we don't know. 2 randomized trials where you either receive nothing or convalescent plasma have reported. One from India, which was quite large, and it reported that the plasma had no effect, but they didn't include any neutralization assays, so we don't know if the plasma that they gave was actually useful. In Spain, they did have a neutralizing assay, and they also reported about a 30% reduction in the death rate. But this was only a small trial with only about 100 or 200 people in it. In the UK, I think we're doing one of the leading trials in the world, actually, and about 5,000 patients have been treated. And the results will be expected probably about the first week of December, and we'll know whether the plasma that we're giving works or not. If successful, I'm afraid we've only got about 2 weeks' supply, so we would encourage anybody who's had the illness to really reply to these letters so that we can benefit people who are sick in the future. Now, there are problems with plasma because it comes from different people. It's extremely variable in its antibody makeup. In fact, there's about 2 or 3 drug companies who now make monoclonal antibodies. So these are antibodies which patients have made in response to getting COVID-19, but instead of taking the antibodies out, we take the actual B cells out and analyze them to actually sequence the antibodies which are made by the B cells, and then they're genetically engineered into other cells so they can be produced, the antibodies can be produced in very large fermenters, a bit like producing beer or wine. And this provides a much more standard supply of antibodies, and this is being used in quite widespread fashion. This was the antibody cocktail, for instance, that President Trump got, that he referred to as a miracle and said he felt much better after receiving it. And we're also giving it in the trial in the UK. The problem is that there aren't enough fermenters in the world to supply a large amount. And at the moment, I think there's only about 50,000 doses of these antibodies available in the world, which isn't nearly enough for the numbers of patients that we have. And then finally, I'm going to come on to T cells. And these are cells that recognize not the virus itself, but whether a human cell is infected with the virus. And if it is, it can kill it. I've got a nice picture here, in fact, of a cytotoxic T cell killing a target cell infected with an influenza virus, but it would be exactly the same for a COVID virus. That's the T cell at the bottom, interrogating the cell, and it's decided it's got a virus, and it's killing it. So there we are. We are also making these T cells from people who've recovered from COVID-19, and we hope to infuse it into sick patients next year to see if this also helps them. So that's a brief summary of what we in the blood transfusion service are using from patients who've had COVID-19 to treat people who come in very ill with the condition. I'd be delighted to answer any questions that you have.